Yes, coming back to Australia, we came back to working on Brahman cattle in northern Australia, and there are some unique things about Brahman cattle which I'll go into during this talk. The animals we sequenced, whether or not they deserve to be legends, remains to be seen, but they certainly are pivotal in the pedigree of the Australian Brahman breed. So let's go back a few thousand years and look at the domestication of cattle. This is a skeleton of a European auroch. There were two subspecies of auroch, a European auroch and a subtropical or tropical auroch. We only have good fossil evidence from the European auroch. The last one died in about 1640. It was owned by the King of Poland, who had a herd. It was basically disease and loss of habitat that got them in the end. So these animals, as you can see, were fairly formidable. They're about 1.5 times the size of a modern domestic animal. So it would have been quite a challenge to domesticate one of these beasts. And there were two domestication centres that we know of. There is evidence that there may be a third in Southeast Asia or southern China. That remains to be seen. But these are the two domestication centres, and one is for the European auroch, is here in modern-day Iraq, and the other one is here in the Indus Valley in what is modern-day Pakistan. There was one domestication event in Iraq, and there seems to have been up to 3 domestication events in the Indus Valley. And that really is reflected in the breed diversifications that we see today. Domestication happened about 10,000 years ago, while the split between the European BOS taurus animals and the subtropical or tropical BOS indicus animals happened around 300,000 years ago. So what happened then? These animals moved with people around the regions. The BOS taurus animals moved into Europe, into Africa, and north of the Himalayas to northern China, and then Korea and across to Japan, which are the basis of the Asian taurine animals, which are quite different to the European taurine animals, things like Wagyu and Hanwu. The BOS indicus cattle, meanwhile, moved south of the Himalayas into southern China and Southeast Asia, into Africa, and then very recently to the Americas, so to North and South America. The major breed in South America, in Brazil, is Nellore cattle, which are called Ongole cattle in India, and the main breed elsewhere is Brahman cattle, which we'll get into. But the result of that is we have 2 subspecies of domestic cattle. We have the BOS taurus animals, and these would be familiar to anybody living in temperate regions, even up to southern Queensland. Hereford, which used to be the king of cattle in the 60s. Everybody had these, but now everybody has these black cattle based on Angus or other breeds. In North America, all the breeds are turning black due to the fact they get an advantage when they sell the animal to the meatworks if it has a black hide. In the subtropical and tropical regions, these animals are important. As I said, in Brazil, Nellore, which are called Ongole cattle in India, and Brahman in Australia, southern US, places like Colombia in South America. The result of that is we have 2 different production systems in Australia. We have a southern system. You can see the difference. The grass is green and there's more of it. So the southern system is running mainly black cattle these days. The temperatures are lower. These animals are very good, productive animals. But if you put one of these animals in a northern environment, no doubt they'd die. So these animals are productive in the north because they don't die. This is a picture I stole from AACO. You can see here these are a composite, what's called a composite animal. They're a mix between Brahman and other taurine breeds to try and boost productivity and meat quality. It's an old photo because you can see a guy on a horse. These days he'd be in a helicopter. He's actually leading the cattle, he's not pushing the cattle. And you would have seen the news recently of today, I think it was, where you saw a helicopter getting the animals out of floodwater around Rockhampton. So things have changed in order to reduce labour costs. So you don't see, you see as many cattle in these extensive regions, but the attempt is to reduce the amount of labour. So the basis of the north-south divide in Australia, and really it doesn't do justice to the north, because the north, as you can see here, is varied in its number of cattle they can run per hectare. The darker the colour, the more animals you can run per hectare. And you can see central Queensland here is quite is a lot better than the areas right up north, where a lot of these big cattle properties are. That run hundreds of thousands of animals on properties the size of a European country. The average farm size is different, 22,000 acres, hectares I should say, in the north, and 5,000 hectares in the south as an average. And it's determined by temperature and climate. So you can see here what's happening. The temperature in those regions up north are far, far hotter, and the BOS indicus animals are tolerant to higher temperatures, higher humidity. And you can see here the rainfall, and the rainfall up here in the north, it's tropical. We've just seen what happens in tropical environments with the cyclone crossing here, lots and lots of rain, whereas the temperate regions have a lot of rainfall, but they're much cooler. So and we also have a wet and a dry season in the north, so in the dry season we have problems with the nutritional value of the pasture declining. The other big thing that's resulted in Brahman cattle being, BOS indicus cattle being preferred in the north is the cattle tick. This photo is obviously not taken in Australia, but cattle ticks are a big, big influence in northern Australia. And there's a lot of work going on in this institute and elsewhere trying to find ways of controlling the cattle tick. It's not a native creature to Australia. It was introduced in 1872. We know when it came. We think we know the name of the barge it came on in 1872 with the importation of 12 cattle. Now this again was probably an attempt to get BOS indicus cattle into northern Australia at the time, but it had devastating effects with the introduction of the cattle tick. The cattle tick has since spread through the wet regions of the tropics all the way down to the border of New South Wales here and Queensland, and is controlled there by the control of cattle movement. We don't have a magic bullet to control cattle ticks yet, so we actually, you've seen the tick gates when you drive down south, and cattle are inspected there to make sure they're not carrying ticks. There are a couple of regions south of that that have ticks, but again, they're controlled by the movement of cattle. So the cattle ticks had a huge effect on productivity. Direct effects were the loss of condition, anemia, death, susceptibility to drought, etc., etc. The animals didn't do as well because you had all these insects. They're not insects, are they? Sucking the blood out of the animals, literally. The market can be compromised because the control of ticks has been the use of chemicals in the past, and that could compromise meat or milk because of residues. The cost of controlling ticks, either with chemicals or any other way, is high, and the cost of the tick-borne diseases is again high, either through deaths and slow growth or the cost of treating the animals in order to control the ticks. So the response to that was the introduction of the Brahman breed, and it was developed in the southern USA from 4 different BOS indicus breeds. This is important because it goes to the diversity we see in Brahman cattle at the moment. They were imported into Australia before the 1930s, but not in any meaningful way until then, and that importation has continued up to 1982 in larger numbers, and then there's still a few cattle coming in from time to time. This is straight off the Australian Brahman Breeders website. It was considered an important addition to the industry. The resistance of these animals to ticks was not really realised until there was a crisis in the price of beef and people couldn't treat the animals with chemical. They couldn't afford it, and they noticed that the Brahman animals really didn't need that treatment. At the time, it transformed the northern beef industry, and it became profitable and highly productive. that's being challenged at the moment because of some of the other attributes of BOS indicus cattle, which I'll get into. So we have the 4 different BOS indicus breeds that was developed in the southern USA, and the Australian Brahmans are unique in that they've been bred up twice. Now, does anybody know what bred up means? There's a couple there who would. So they didn't have many animals to start with, so they outcrossed them to BOS taurus cows, crossed the calves back to the Brahman bulls in order to get the numbers up. Once they were about 15/16 BOS indicus or Brahman, then the herd was closed and the breeding was just within those animals. Then they were imported into Australia and the same thing happened again. So in the US it was across, I believe, Angus, and in Australia it was Shorthorn and Hereford that they were put across in order to get the numbers up. The result of that is that the Australian Brahmans actually have about 7 to 10% BOS taurus content in their genome. So they're a bit of a hybrid, really. They look very BOS indicus, but they have this underlying bit of BOS taurus genome. That, in fact, may result in some advantageous production traits because, as I've said previously, the BOS taurus animals are more productive, their meat quality is better, etc. But we're always trying to balance productivity against tropical adaptation. It's no good increasing the productivity of the animal through genetics if you lose the animal because it's not adapted to a tropical environment. So these two things are always in tension when you're trying to breed animals. That's been addressed by and large by crossbreeding, which I'll get into a little later. But the Brahman cattle are by and large very good in terms of their adaptation to a tropical environment. These are the 4 breeds of BOS indicus cattle that we use to make Brahman. We have Gyr or Gyr, which are a dairy breed of BOS indicus. There's a lot of these in Brazil. They were used because they wanted to get the milk production up on the cows to raise better calves. We have Gujarat, Krishna cattle, and what in Brazil, this is a Nellore, but it would have been a Ongole animal from India. So let's have a look at profitability. Tropical adaptation survive in harsh environments. So it's no good having a really genetically superior animal if it's not tropically adapted, because it will probably die. And in fact, that has happened with some things that have been tried in the north as the animals haven't survived. We have low input systems in the north. We have these very extensive management systems. You saw the number of cattle being herded in that slide. So many, many animals, but you don't see them very often. They have to forage over large distances, and the tropically adapted cattle are good at that. And we have natural pasture-based production systems for these animals. So we can say that we've not messed with them too much, and that can be an advantage in terms of the consumer. We have economies of scale. Any improvement we make in animals is multiplied over very large numbers. And we have, in Australia, the involvement of very large companies in the cattle enterprises. And this is, again, probably unique in Australia in that we have these very large pastoral companies. But there are some negatives as well. Zebu, which is another name for BOS indicus cattle, grow more slowly. So we have another year before you get these animals to market. So remember, if you're farming cattle, you've got a cash flow going on here. So if it's another year to get an animal to market, that's another year you're not getting paid. We have wet and dry season variability. The animals lose weight in the dry season because the nutritional value of the feed goes down. We have late puberty and long postpartum and estrus interval in BOS indicus cattle. This is again something that's due to tropical adaptation. It's not good for the animals to be too fecund, because if they have to support a calf in hard times, that can have very detrimental effect to the cow. However, for the producer, it means 2 extra years before they make money. And they need to have 3 calves from a cow before they're making any money. So you're waiting another 2 years there and another 1 year up there for them to reach market. Zebu cattle generally have lower meat quality, and that can be translated into toughness. The Zebu beef is generally more chewy by a large degree than the BOS taurus beef. But there's a wide variation in that. You can get some quite palatable Zebu beef. We have, and you always hear about live export markets. And these have become very volatile through political and other reasons. We have now countries arbitrarily setting quotas, and they can be changed at very short notice, meaning that supply may be compromised and not meet the demand of the country importing the cattle. And we have more and more public scrutiny on animal welfare of the animals that leave our shores and end up in China or Indonesia or Vietnam or somewhere else. I think that's a good thing, and I think the industry should embrace that, because if they're on top of it, then the public criticism will largely be answered. So the result of all that is, although we have the largest number of cattle in northern Australia, and I'm drawing an arbitrary line here at the border of Queensland, say, the largest value resides in the south of Australia. So the smaller number of animals in the southern region, because they're BOS taurus with all the meat quality advantages and the faster growth and the more fertility, we have a higher value down there. So the challenge is to raise the productivity of the animals so that we can bring the northern Australian industry to a higher value. So what are we going to do? We need to remain globally competitive against our international competitors. Now, at the moment, we have a bit of hiatus in the competitive sphere, because Brazil has been compromised with some of their health and safety standards through their packing plants, through their abattoirs, and has been, that's compromised their markets, especially into China. And we have in India a ban on buffalo slaughter in the major province that provides buffalo meat. So the Australian producer at the moment is looking at good times, at least in the short term, because there's an undersupply of beef into the countries that are buying our beef. The demand for beef is increasing in countries like China, Southeast Asia, Indonesia, because as people get some more money in their pocket, they want more protein in their diet, and the sales of beef go up. That's contrary to what's happening domestically, where the sales of beef are either flat or declining a little bit. So we need to increase our productivity. That can be nutrition. Fertility is the big one, because that's directly relevant to the producer. Growth, and improve the quality. And that's been done largely through crossbreeding. So you get your BOS indicus animal, you put it across a BOS taurus cow. That's a fairly blunt instrument genetically, but it's been very, very effective. And there's a lot of opportunity, I believe, to make this more sophisticated and get even better results from crossbreeding. And of course, we have breed improvement within the breed, which can impact the crossbreeding as well. So a lot of these issues, not all of them, but a lot of the issues can be addressed by genetic improvement. In Northern Australia, there has been little genetic improvement achieved in the last few decades. And there are reasons for that. That's not a criticism. There are good reasons that genetic improvement is difficult when you only see the animals once a year. You can't measure the birth dates, you can't measure the growth rate accurately, etc. But that's the production system we have. Again, crossbreeding's been used effectively, and again, the reason that we have a problem or a challenge is that the performance data is difficult to collect. So the Australian Brahman Breeders Association, to their credit, have taken moves to address some of these issues, as some of the large pastoral companies have done, and that is to create a nucleus herd of animals where you can measure the traits intensively. The unfortunate bit is these, if we're going to apply genomics technology, need to be very big. We need to have thousands of animals in those herds, and that's not been achieved yet. But we do have benchmarking, progeny testing on these traits, And there are some international collaborations with the USA, South Africa and Namibia to pool data in order to improve things in terms of traditional quantitative genetics, if not molecular genetics or genomics. So I'm going to say here that unashamedly that genomics can be an answer to some of these challenges. All our genomic information in terms of the sequence is based on this animal, which is a Hereford. That's northern Montana. I don't know whether any of you have been to the east part of northern Montana. It's the Badlands. It's where Custer met his doom, and I don't know why he was fighting over that land. It's awful. But anyway, that's Dominette. She's the animal we sequenced and published in 2009 at the cost of $50 million US. So we now have all these tools in our toolbox: genotyping, sequencing. We can do genomic evaluation if we have enough data, and we can find causal mutations. But again, all based on the sequence of this cow, which is the wrong subspecies. And I'll just give you an example of something here. This is a study we did in Brazil on Nellore cattle, and we're looking at age at first calving, which is a measure of age at puberty. Okay, we've done a very similar study in Australia, and our measurement was a bit more sophisticated than this. We ultrasounded the animals and looked at the first corpus luteum, which is the first time the heifer cycles, and that was the measure of age at puberty. But anyway, we find this nice big peak on chromosome 14 here. And it's unusual for a multi-gene trait because it may explain as much as 8 to 10% of the variation of the trait. Usually, when we find peaks like this in multi-gene traits, we're only explaining 1% or less of the variation, but this is a big one. And when we look further into this, there was a candidate gene, Marina Fortes, in the BCRC work she did, found the PLAG1 mutation, which had been described in dairy cattle as a mutation that affected conformation or size. And we looked at the region around there, and what we found was that the advantageous allele in PLAG1 was actually from BOS taurus. So it's a little bit of BOS taurus genome that's found its way into BOS indicus, into Brahman. And the other interesting thing is, this is Brahman, and if we look at linkage disequilibrium, just in the region around that PLAG1 mutation, linkage disequilibrium is a measure of whether or not two bits of variation in on the chromosome are going to segregate together or they're going to split due to recombination. And we can see a very large part. The darker the colour, the more linkage disequilibrium. There's this big block in Brahman of linkage disequilibrium of the BOS taurus bit of the genome. In Nellore, it's much smaller, which one interpretation of that could be that that bit of BOS taurus came across into Nellore a lot long before it came across into Brahman. That's one explanation. There are many others, but let's go with that for now. So let's look at these animals in terms of their breed composition. This is a principal component analysis, and it basically looks at, in this case, genotypes and groups individuals on similarity. It doesn't tell you why they're similar, it just tells you whether or not they're similar, and we can do that in this case in 2 dimensions. And we can see here Nellore are very compact. Nellore have about 1% BOS taurus. They didn't have that breeding up process, but a bit of BOS taurus has snuck in. It may have snuck in even before domestication or in the early days of breed formation when it was still in India or Pakistan. Brahman, on the other hand, are somewhat spread out. So some of them have more BOS taurus than others, because over here we have the 2 BOS taurus breeds, Shorthorn and Angus, and we can see Brahman stretching out towards these. So these animals on this end of the scale have more BOS taurus genome than BOS indicus. There's a large amount of breed diversity in the Brahmans, and this is again because they use 4 different breeds of BOS indicus cattle to form the Brahman breed. And we can see here in red we have Brahman. This is each chromosome along here, and we can see the number of variants we find in the Brahman compared to that of Angus. And we can see here it's at least double the variation we find in the Angus breed. And again, that's reflected again in the effective population size of the breed. Effective population size means that how many genomes do you have in the population? So although there are very many Black Angus cattle in the world, because of the high use of artificial insemination, effectively there are only 150 animals there. Whereas we see in Brahman, there are 3 to 4 times more. These animals here are crossbred animals, a cross between a Bos taurus and a Bos indicus, and we can see here Droughtmaster especially have a very high population size, effective population size. Santa Gertrudis, which have been bred to be more and more alike, you'd expect them to be more, inbred's the wrong word, but less diversity. You can see that's actually happened, but they're still well above Angus. Holstein, the black and white dairy breed, is the lowest. They're about 100, effective population size of 100 in the 13 million Holstein cattle that are kicking around the world. And again, this just reinforces that we can see the crossbred animals here sitting between the Bos taurus and the Bos indicus animals. Interestingly enough, we can see here some of the Brahman animals are drifting into the composite animals, and that'll be even more evident in this. This is those two crossbreds, the Droughtmaster, which is bred to be more diverse, and the Santa Gertrudis, which is bred to be less diverse. And we can see here the Brahman cattle are drifting over, and we have these animals which are crossbreds between Brahman and something else drifting into the Droughtmaster space. So putting all that together, we believe that we need a reference sequence for Bos indicus cattle. But using the Bos taurus reference sequence is leading us astray in many places. And indeed, the Bos taurus reference at the moment has many areas in it that are not very well assembled, which makes life even more difficult. And further to that, because of the unique genetic history of Australian Brahman, we should do a reference sequence for that animal. That would seem to be the animal that we should do a reference sequence for in order to be relevant to the local industry. So our first step in that was short read sequencing. And short read sequencing is sequencing of about 100 bases of genome. And we do that so we get about, on average, 12-fold depth of coverage of each animal. So each animal has 3 billion bases, and we're doing it in 100 base pair chunks. So you can't build a reference out of that, but you can look at the population diversity. And we did that, and we chose 100 influential sires, and we found semen, stored semen from those sires, and the oldest one we found was born in 1953. So that semen had been stored since about 1954, 1955. It's remarkable. Brian Burns helped a lot with that, as did the Australian Brahman Breeders Association. So far, we've done 50 at an average 12-fold depth, so 3 billion times 12. 36 billion. We have included 3 polled animals. Polled is the lack of horns, and that's very important in Australia because we want to do away with horns on cattle. The polled gene in Brahman is very rare. It's getting more and more prevalent as people are working towards increasing the frequency of that allele in the gene so that we can end up with a population of animals that don't have horns, which is a lot better for the people handling the cattle. They're a lot less likely to get hurt, and the animals are less likely to hurt each other. This is going to mean much to anybody. These are the animals we've sequenced. We don't have the data back for the polled animals yet. That's on a hard drive between Hong Kong and Australia somewhere at the moment. But anyway, we captured most of the studs. And here we go. This is, again, the principal component analysis, but now just with the Brahmins. And a couple of interesting things here. One is the population of Brahmins is actually splitting. most of the animals are here. You can't, it doesn't look like that, but they're all on top of one another. So the majority of animals here, these are far fewer animals actually here. But our historic animals sit somewhere in the middle. So as time's gone on, we've had this split. And again, principal component analysis doesn't tell you why. We can hypothesize that this might be due to selection on fertility, for example, in the population, but we really, at this stage, don't know. So why have we chosen these old animals to sequence in terms of diversity? And the reason is we may have an animal up here. He might be an animal born in the 70s or something, and he has a mutation here, a G to T mutation. His sons, some will inherit the T and some won't. So it's entirely possible, but by the time you get down a few generations, that the T allele might become quite rare and you may miss it in your sequencing. So the further back you can go, the more likely you are to pick up all the mutations that are segregating in the population. With the 46 animals we've sequenced so far, we're only capturing about 17% of the variation in the breed. So we still have a long way to go. We're hoping that with a wise choice of the next 50 animals, we can increase that significantly. In those 46 animals, if we just look at the single nucleotide polymorphisms, we have just over 36 million variants. That's a very, very large number of variants in the breed. Of those, if we look at things that might affect the function of a gene, we can go here and we can go, what's in coding sequence? 69,000 of those 36 million. And which are the missense mutations? And this comes down to a small percentage, 83,000. And then we have a small number of things where a stop codon's been put into a gene, so that would effectively silence that gene. And we have things that may be in control regions of genes in the untranslated regions of the gene, for example. So lots and lots of variation there, lots of scope for identifying genes that may affect performance. Now, the cautionary note there is that there's been an initiative to knock out, to silence every gene in the mouse genome, and most of them have no effect. And when I mentioned this to a bioinformatician, he said, yes, but have you ever taken the animal to the opera? It may drop dead. So environment plays a big factor in this, and because we don't see any effect of the gene doesn't mean there's not one. So we've looked at all those 46 animals, and we've done what's called heat maps based on FST, which is a measure of how much the bit of genome we measure, and you can see here the size of the chunks, is like a taurus gene, and how much it's like an indicus gene. And the redder they are, the more they're like a taurus gene or segment of chromosome. We have the oldest animal up here, 1953, and the youngest one down here, which I think is around 2005. This is chromosome 23. Chromosome 23 is important because it has the major histocompatibility complex on chromosome 23, and it's right here. So this is your adaptive immunity. And you can see here on first glance, you think, oh, it's captured a lot of BOS taurus. Now, there's a nice explanation for that. In that particular part of the genome, you want as much variation as you can because you want to be able to react to whatever pathogen's affecting you. The rider on this, and we'll just look at it like this. This is, if it's blue, it's more BOS taurus-like, and if it dips down, it's more BOS taurus-like, and we can see here this region. But it's a very poorly assembled region of the genome. And when you look back at this, you can see that really there's been nothing happening over time. So another explanation for this might be we don't have a very well assembled bit of the genome. When we run it through the computer program, it doesn't know where to put it, and it's thrown it into taurus because it doesn't know where else to put it. So what may well be artifactual. We have another region just looks like this, which is a detoxification region, which again makes sense when you think about it, but may just be because it's another poorly assembled part of the genome, because it's an adaptive part of the genome which changes quickly. Let's get back to this age at first calving, or puberty. As I said, chromosome 14, we get this big peak. What happens to our BOS taurus content on chromosome 14? And we can see here, this is a very different picture. This, we can see here, lots and lots of animals have this BOS taurus segment retained. More than that, it's increasing over time. Again, oldest animal here, youngest animal here. It's increasing over time, and we can see there's crossover events going on here, which puts it back in the BOS indicus part for this part of the chromosome here, but it retains this bit here. So this is much more convincing, and we can see here if we plot, and in this case we split it between old and young animals arbitrarily, admittedly, because if we take the averages over the whole lot of animals, back to 1953, you don't see any effect. But we can see in the younger animals, certainly, there's a bigger incidence, higher incidence of this bit of genome being retained. And indeed, that's what you see in the younger animals. We can see the BOS taurus bit being retained. And it's very, very nicely defined in that region. And if we look at that region, and you're not going to be able to read this, so I can make no apologies for that, because this is what the database spits out. We see this is PLAG1, that gene that was postulated as being the causal gene for the age of puberty or the conformation. But there are two other genes. There's one called PENK, and there's XKR4, sounds like a model of a falcon to me, that also have been implicated in productivity and reproduction. So there may well be a complex of genes in this region that are affecting the same trait. It is the only region we see in the genome where this is happening. And again, you might postulate what's been selected in Brahman cattle when we've had this big extensive production system. Now, the industry has selected animals on size. We know PLAG1 is implicated in size of animals in conformation, but also if it's puberty, only the animals that have a calf will go into the next generation. So animals that are more likely to have a calf are going to contribute more to the next generation than animals that miss their first calf or miss their second calf. So that's really no human intervention involved really in that. And that's a way that could happen to increase the BOS taurus content in this. But I think it's significant that we don't see anywhere else in the genome, we don't see this effect happening. So we want to do a reference sequence. We have selected our cow, L. Rose Naomi, 3492. We sampled her, so she's no more, in October last year, and we have blood and multiple tissues from that animal. We will prepare DNA from lung, and that's supposed to be happening any day now, for sequencing. Now, we're not going to use that short read sequencing. We're going to use a technology called PacBio, which instead of 100 base pair reads of sequence gives you 10,000 base pair reads of sequence, which are far easier to put together into a whole genome. That will be done in one of 4 labs, depending on what they quote. And then we're using a technology called Dovetail to help put all that together into a sequence. This has been demonstrated, certainly in goat, to give you a very, very, very good reference genome sequence, far better than anything before. Our project has a 2-year time frame, based mainly on the way the money flows, not on how quickly we can do it here. And then we're going to map all those short reads onto the reference sequence to get a clearer picture of what's going on in time in terms of bits of BOS taurus genome retained. There she is. She was a very productive cow and well connected in the pedigree, so a bit of a blue blood. So what are the outcomes of that? We'll end up with all of the variation in Bos taurus cattle in Australia. That's our aim, or as much of it as we can. It's probably a bit ambitious to say all of the variation. At the moment, we have 36 million SNPs, and if we include the insertion deletions in the genome, I think it comes to around 39 million variants in the genome. And remember, that's only 70% of the population. More importantly, we'll have long-range structural variation because Bos indicus and Bos taurus have been separated long enough for bits of the chromosomes to have inversions, etc. Small pieces, but it's going to be there. So we're going to be able to identify all of those so that we don't get the order of genes wrong when we're walking down the chromosome to identify a causal mutation. Hopefully, we'll get a publication like the sequence publication. Now, I note, you should have noticed that this is not the animal we sequenced. When it was published in Science, they decided to put a pretty one on the cover. And again, we have a pretty Brahman here. Maybe that might be on the cover. So the impact of that is improve our predictions of fertility meat quality on the next SNP chip, and that would feed into breed plan in Australia, which is where the genetic evaluation is done, be it traditional or genomic. Low cost and accurate sequencing of bull calves. This is mainly through a technique called imputation, where you can genotype the animal on, say, 50,000 SNPs, which is much less expensive than the sequencing, and then deduce what the sequence is from that. And that can be very accurate if you have enough animals sequenced. And initially, or well, in the end, sorry, greater genetic gain in the northern cattle populations. So with that, and Matty, I apologise, I left your name off this. Ben Hayes, Ross, who's right here, who's done most of the computer work for us. Brian Burns, who helped with our selection of bulls, ran around the countryside finding the semen in the tanks. Russell Lyons at the genotyping lab out at Gatton. Bronwyn Venus, who's helped us technically. And the Australian Brahman Breeders Association, who have been really great in their support of this. So I'll leave it at that, and happy to answer any questions.